Good evening, everybody. I first want to thank all of those who have attended tonight. My name's Shannon Strader, and I was this past year's RFC research representative. I am also the creator and moderator for the AAP Orthobiologics and Regen Med series. The goal for the webinar is to provide comprehensive education for physicians in training and physiatrists interested in regenerative rehabilitation while reducing stigma, misinformation, and encouraging responsible advancements for the regenerative field. If during the session you have a question, please write it in the chat box, and we will answer as many as time permits. Also, at the end, we will be doing a Q&A.
It gives me great pleasure in introducing our brilliant panelists. Dr. Linda Jones is a physical therapist and scientist who is currently a faculty member at Thomas Jefferson University and consultant to biotechnology companies, universities, and nonprofits, including the Christopher and Dana Reeve Foundation and the International Spinal Research Trust. After focusing on physical therapy interventions in the intensive care unit, she managed the first cell-based trial in spinal cord injury, followed by the first study utilizing cells derived from embryonic stem cells in the United States. She is co-chair of the Spinal Cord Injury Outcomes Partnership Endeavor, the chair of the research committee of the American Spinal Injury Association, and has served on the National Institute of Neurological Disorders and Stroke. Dr. Edward Worth is a physician scientist and is currently the chief medical officer for Aspen Neuroscience. Dr. Worth is leading the company's upcoming clinical programs studying its first cell therapy candidates to treat Parkinson's disease. With more than 25 years of experience in surgical applications and clinical trials in the cell therapy space, he is one of the neurological experts in translating regenerative cell science into clinical programs. His work at the University of Florida included leading the team that performed the first to transplant human embryonic stem cells into the spinal cord in the United States. He next served as a chief medical officer at Lineage Cell Therapeutics in 2019. Dr. Stephen McKenna is a clinical professor in the Department of Neurosurgery at Stanford University School of Medicine. He is a founding member of the Stanford Partnership for Spinal Cord Injury and Repair. He is currently the chief of the Respiratory Rehabilitation Unit, as well as the site director of Advanced SCI Medicine Fellowship at Santa Clara Valley Medical Centers. He has led first-in-human clinical trials ranging from vaccines for HIV to stem cell-derived oligodendrocyte progenitor cell-based therapies for spinal cord injury. Dr. McKenna is the chairman of the respiratory section of the International Standards to Document Autonomic Functioning Following SCI, and co-chair of the Asia Research Education Committee. Finally, but not least, Ms. McCulley joined Lineage as Chief Executive Officer in September of 2018. He previously worked at the University of California, San Diego, Department of Technology, Transfer, and Intellectual Property Service, and he conducted drug development and research for Neurocrine Biosciences. He has also served on the board of Orphagen Pharmaceuticals since 2017. He has more than 25 years of business and scientific experience in the life sciences industry. Thank you again for speaking to all of us tonight.
Thanks for that introduction, Andrew. So on behalf of all of our speakers today, I'd like to thank AAP for the invitation to speak. This session is sponsored by the Research Committee of the American Spinal Injury Association. However, there is no endorsement of any products or services discussed in this presentation. I'd also like to kind of frame this talk in the context of the intent of the talk today is really to discuss advances in clinical trials using cells that are derived from embryonic stem cells. And to illustrate some of this progress, we're going to talk about specific studies that used oligodendrocyte progenitor cells for individuals with spinal cord injury and thoracic and cervical injury. In terms of our speakers today, thank you very much for the introduction. As highlighted, myself and Steve and Ed are in other roles and are not employees of Lineage, and Brian is the CSO of Lineage. And again, we really appreciate this invitation to speak to you today about advances in stem cell research. Lineage is a publicly traded company, and as such, any forward-looking statements are based upon current knowledge and expectations that may prove incorrect. So I'm going to start with talking about the journey and where the oligodendrocyte journey began, and I'll shorten that to OPCs because it's always a mouthful to say oligodendrocyte progenitor cells. So the journey began in 1999 when Geron Corporation began the development of the embryonic stem cell line that we used in these clinical trials. And Geron first received FDA investigational new drug clearance in 2009. And this was the first ever clinical trial using cells derived from embryonic stem cells in the US for any indication, which happened to be in spinal cord injury. The initial clinical trial was in thoracic spinal cord injury, which was discontinued when the company deprioritized this particular arm of investigation within the company, which was not related to any findings in the clinical trial. And the trial was discontinued in 2011 after a sample size of 5 individuals enrolled in the study. The asset was then acquired by Asterias Biotherapeutics in 2013, and they executed a clinical trial in cervical spinal cord injury with a sample size of 25 individuals. Lineage Cell Therapeutics then acquired the asset in 2019. And since that time, they have focused on improving manufacturing of the cell product and the device used to deliver the cells. And they plan to test the delivery device in 2022 and start a comparative trial in 2023. And you'll hear more about that later in the presentation. So I'm now going to talk about the thoracic trial. And I don't really need to go over some of this background for this audience because many of you are well-versed in spinal cord injury. But just to provide a little bit of context, the incidence of spinal cord injury in the United States, as determined by the spinal cord injury model systems, is approximately 18,000 new cases per year, with an average prevalence estimated to be 294,000 cases per year with a range of 249 to 363. The primary cause of spinal cord injury is vehicular, either any type of motor vehicle from cars to motorcycles, et cetera. And the second major cause of spinal cord injury is from falls. And the incidence of falls has increased of late, given the older aging of the population. In terms of levels of injury, it's not on this slide, but about 60% of individuals with spinal cord injury have an injury resulting in tetraplegia and 40% paraplegia. So spinal cord injury is a high burden and an unmet need, and it's really unique in terms of CNS dysfunction in that an individual, unless they have a concomitant brain injury, is cognitively intact, but may have very significant impairments in function. And these impairments range from loss of mobility to intractable neuropathic pain. These individuals can have frequent infections requiring hospitalization, bowel and bladder and sexual dysfunction, and for high-level spinal cord injury, ventilator dependence. And these can result in a higher incidence of depression and a shortened life expectancy compared to the population without spinal cord injury. And these impairments result in a significant burden for patients and caregivers, wherein 67% of patients in the United States are unemployed 10 years post-injury, and lifetime costs can reach $5 million. But I think that these are all facts and figures and words on a paper, but the right-hand panel really illustrates some of the challenges that people living with spinal cord injury and other disabilities encounter on a day-to-day basis. This is likely a staircase coming out of a subway. And for some, including those in wheelchairs, this can be as great a hurdle as Mount Everest. So as mentioned, loss of movement is the primary feature of spinal cord injury. And as you know, higher level injuries in the cervical spine result in greater, particularly in the cervical spine, result in greater impairments. And gains in motor function, particularly in the cervical spine, can result in the difference between independence in a given activity and requiring assistance in certain activities. And the goal of Lineage's current direction is to improve upper extremity function, increasing independence and quality of life. So the Lineage technology platform, these are allogeneic cell transplants, meaning they did not come from the individual who receives the transplant. The product begins in a frozen vial of pluripotent stem cells, and these pluripotent cells can become any cell type in the body. However, these cells are differentiated using Lineage's proprietary process into, as mentioned, oligodendrocyte progenitor cells, or OPCs. Throughout this process, no intentional alterations are made to the cell's DNA, and there is a rigorous in-house manufacturing process that allows for scalability. So as you well know, oligodendrocytes are the cells, the precursors to, oligodendrocyte progenitor cells are the precursors to oligodendrocytes. And these are the myelinating cells in the central nervous system, and myelin is essential for proper signal transmission in axons. So what do these cells do? Where there are three major activities that have been identified for these cells, and one I just mentioned, which is as myelinating cells. And all of the panels that you can see here are in the rat model. So on the left, you can see that the cells wrap post-axon and form compact myelin sheaths, which you can see with the red arrow in the panel on your left. But these cells also produce neurotrophic factors, which stimulate neurite outgrowth, which can be seen on the upper right-hand panel here. And they also stimulate neovascularization. In addition, following spinal cord injury, and again, this is in a rodent model, as illustrated here, following spinal cord injury, there is a cavitation at the lesion site. And as you can see on the right-hand panel, there is much less cavitation and evidence of tissue within this cavity. And this has also been found in the studies in humans with spinal cord injury using OPC1. So the product is a cryopreserved, as I mentioned, allogeneic cell product, and it is derived from an NIH-registered human embryonic stem cell line, the H1 line. The vials contain oligodendrocyte progenitor cells, as mentioned. They also contain neural progenitors, as well as some mature neural cells and other rare characterized cell types. I've already mentioned some of the functions of these cells related to neurotrophic factors, vascularization, myelination. During the thoracic trial, there was a procedure after there was a preparation step when the vials, from the vials to get to the patient. But currently, this is an off-the-shelf process, which will be discussed later in this presentation. And the first indication for these cells is in spinal cord injury. So these images are fantastic images of how the cells are delivered in the operating room. Although these images are from the cervical study, they are quite relevant with the same procedure for the thoracic trial. And the injections were performed, as you can see here very nicely on the left, using what was called a syringe positioning device. And this device was mounted to the table in the operating room. And I'm not going to go into detail because Edworth will discuss this in more detail later. And you can see here in this lower right panel an actual injection. And these cells were injected intraparenchymally into the spinal cord lesion. And in the thoracic study, this consisted of a single 50 microliter injection of 2 million cells. In the cervical study, the injections were 2 million and 10 million, and then 2 injections for a dose of 20 million doses, or 20 million cells. So an overview of the thoracic study, this was an open label trial. 7 sites were used to recruit individuals to the study, and the intended sample size was 8 to 10 subjects. in individuals with motor and sensory complete spinal cord injury, otherwise known as Asia Impairment Scale A, with neurologic levels from T3 to T11. As mentioned, the cells in the thoracic study, 2 million cells, and the transplant occurred 7 to 14 days following spinal cord injury. So as you have all taken care of individuals with acute spinal cord injury, this is a tough time window logistically. And individuals in this study, because this was an allogeneic cell product, were given temporary immunosuppression. The primary assessment in this study, as a phase 1 study, is safety, but there were 3 different areas that were identified in relation to safety. One was the cells themselves. The second was the surgical procedure to deliver the cells. And the third was the concomitant immunosuppression administered. The secondary assessments were neurological function as measured by sensory and motor scores. And for this, we used the standard neurologic exam for spinal cord injury, the international standards for the neurological classification of spinal cord injury, otherwise known as INSCI, which doesn't flow off the tongue very well. As you can see in the panel below, this is the study schema. So the screening period occurred up to 3 days prior to the cell injection, followed by a baseline period to confirm neurologic level of injury and other key criteria. The injection was given on day 0, and at this time, immunosuppression was begun. Immunosuppression was tapered was discontinued at day 60 after a tapering process, and the immunosuppression was using tacrolimus. The first study protocol covered the time period from screening through 1 year, where there were frequent and intensive visits and data collection. From the period beyond 1 year to 5 years and up to 15 years, this was a second protocol. Data from years 2 through 5 were collected in in-person visits, one visit a year, and up to 15 years by phone follow-up. And as you can see, there were many MRIs throughout this period because due to the cell-based nature of this product, it was important to have adequate visualization follow-up of the spinal cord. In fact, there are 9 MRIs in this in the protocol. So as mentioned, this is the thoracic study, and I show this slide for a number of reasons. So this is the INSCI exam, the standard exam for neurological function following spinal cord injury, as mentioned previously. And what you can see highlighted here are the thoracic segments that were part of the inclusion criteria in this study. And a couple key points here. One is that for a clinical trial that is a high-risk study, the FDA will mandate that the study is initiated in thoracic spinal cord injury and incomplete or very severe spinal cord injury. And the reason for this is something I alluded to earlier, which is that a loss of function in cervical segments can mean the difference between independence and dependence. But a loss of function in the thoracic segments has less impact on function. The challenging thing about running a trial with thoracic spinal cord injury is that when you look at effectiveness, you're only able to look at, using the INSCI exam, at the dermatomal segments represented here, so which are less reliable to assess than motor function. So looking at the demographics of the thoracic study, the age range was 21 to 32 years of age. 4 of the subjects were male, 1 was female, and the levels of injury spanned T3 to T8. The cause of injury in 4 subjects was vehicular, with one with a fall. And these demographics are typical of spinal cord injury with current data collection. There were 3 sites that ended up enrolling subjects, the Shepherd Center, Rehab Institute of Chicago, and Stanford in conjunction with Santa Clara Valley Medical Center. This is a consort diagram, and this diagram is actually lifted from a publication. So the thoracic publication was just released as a pre-pub this week, and actually will be available hopefully within days as an open access publication. This is in the Journal of Neurosurgery Spine, and the lead author is Dr. McKenna. So you can find this now, but hopefully within the next several days, it will be available open access. And the cervical manuscript is currently under review. So this is the consort diagram from the Thoracic Study, in which 11 patients were screened for participation. This does not include pre-screening procedures for, or I shouldn't say procedures, but pre-screening for individuals that may be interested where they had to meet the inclusion-exclusion criteria. But of the individuals who participated in the informed consent process, 11 were screened, 6 failed screening, 4 for metal artifact from MRI. As mentioned, due to the nature of the cell-based product, it was important that the area of injection could be visualized on imaging. 1 subject screen failed because their neurologic level was found to be at T1 outside of the segments included in this trial, and 1 had increased liver function tests beyond that which was allowed in the inclusion criteria. Of the 5 subjects who received the cells, all remained in the clinical trial and have completed long-term follow-up up to 10 years, which is presented in the paper that I mentioned. So looking at the adverse events in this clinical trial, in the thoracic trial, there were, this table is in the publication, and the table presents the adverse events by system organ class. There were 174 adverse events, 25 of which were determined to be possibly related adverse events. And as mentioned earlier, there were 3 areas that we considered to be 3 areas of possible adverse events. One was the cells, in which no adverse events were thought to be possibly related. The second was the surgical injection procedure, where 9 events were determined to be possibly related to the procedure to inject the cells. And the highest number was 16 adverse events that were determined to be possibly related to immunosuppression. And as you can see, these are common side effects of tacrolimus. Of the serious adverse events, there were 4, again, determined to be unrelated to the cells, the procedure, or immunosuppression. The 4 adverse events occurred in 3 individuals in the study: pyelonephritis, urinary tract infection, increased autonomic dysreflexia, and a mood disorder. So looking at the secondary outcome of neurological consequences or neurological outcomes, what you can see on the right, and again, this is in the publication, are the assessment of the sensory dermatomes. And vertically, you can see the subject numbers displayed here, 1 through 5. And from left to right, this is the baseline exam, and this is the year 5 exam. And so green is intact sensory function, yellow is impaired, and orange is absent. And what you can see here is that there are small changes in sensory function. For example, in subject 1, where they had improved sensory function in 1 dermatome, and in others, they actually had some decreased sensory function. But all of this is actually within the context or within the margin of error in a sensory exam. And I'll come back to that in a minute. In terms of motor scores, there were no changes in either upper or lower extremity motor scores. Upper extremity motor scores remained at 50, and there was no motor improvement in the lower extremities. As mentioned, individuals recruited to this trial had motor and sensory complete spinal cord injury, also known as American Spinal Injury Association Impairment Scale A, or AISA. In terms of changes in neurologic level of injury, which were in this study, as mentioned, because this was in thoracic spine and reliant on sensory dermatomal testing, 2 individuals had no change in neurologic level of injury, 2 had 1 level of improvement, and 1 had a 1 level loss. But again, as mentioned, this is in the range of variability in the examination. So now I'm going to turn it over to Dr. McKenna, who's going to discuss findings from the cervical study.
Great. Can everybody hear me?
Yes.
Yes.
Yeah. Okay, good, good. Well, thanks for the lead into this, Linda. just to put a little bit of context, Linda talked about the first 5 individuals who were in the thoracic trial. I'm going to talk about 25 individuals who participated in the cervical trial. And as Linda mentioned, I also am not affiliated with Lineage. I've never been actually corporately sponsored by any organization, and have really had the pleasure of working in the public sector through Stanford and at the public hospital in Santa Clara for the last 22 years, something like that, give or take. So with that, Linda had addressed a slide that's very similar. I wanted to start with something that kind of gives you a graphical idea of where the cervical trial sort of moves. So moving up from the thoracic, this trial enrolled individuals who had C4 to C7 level injuries. And I'm sure everybody on this call is familiar with this, but I think it's relevant to understand that one of the sort of goals leading up to this was to actually start to, even though it's still a safety trial, move towards evidence of efficacy. And if you can stay on this one for a second, Linda, if you went back. Yeah, you can see this on the slide. I'm not sure, depending on the size of your screen, but remember C5 would give you elbow flexors, C6 would give you wrist flexors, and wrist extensors, and C7 would give you elbow extensors. So, these are muscle groups that are easily measured and quantified. And you can go to the next slide, Linda. Moving beyond just what the numbers would show, which is, I think, a bit of a pitfall that we can get stuck in with the INSCI exam, is that there's something really practically important about getting these types of levels back. So if you were to look at the left side of this graph, this is sort of derived from some work that Gail Whitenick had done in the end of 1999, is the red is areas where people would be total assist for activities, and you can just go down the side of transfers, eating, dressing. As you move into the yellow, you're having partial assist, and as you move to the green, you're independent. And I think this really graphically starts to give people an idea what kind of improvement you would have in your quality of life and your independence with 2 levels. I think one more click is going to put an arrow on this, Linda. So if you went from C4, which is the highest level enrolled in this trial, to C6, you'd be moving from an area of total independence with the majority of activities of daily living to an area where you would need some assist. And then if you went from C5 to a C7, C8, you'd be moving to a very independent lifestyle. So these are kind of characterizing the types of improvements that you might see with 2 levels. We can go to the next slide. Now, as I mentioned, there are 25 individuals in this clinical trial. There's some complexity to how they were enrolled that this slide helps to simplify. The trial started, obviously with cohort 1. That completes a group of 3 individuals with 2 million cells, and that established, in agreement with the FDA, the surgical procedural safety for the trial. Then moving into the next 22 patients, now we're starting to talk about getting dose escalation and potentially moving into what we're calling the intention to treat analysis. The first group of people in that group received 10 million cells, and those were injected, as Linda had mentioned, intraparenchymally at the inferior margin of the lesion. As you move to cohort 3, the dose increases to 20 million cells, and the way that that's done is by having 2 injections performed within the lesion at the rostral and caudal ends, with a goal to have 5 millimeters of separation between them. Those were all initially in AISAs, the most complete injuries. Then moving further in the trial, AISBs were enrolled in cohort 4, now with 10 million cells, and in cohort 5 with 20 million cells. So that's a layout of the 25 individuals in the cervical trial. You can go to the next one, Linda. One of the challenges of trying to run trials of that complexity is you start to run into some asterisks, and I just wanted to highlight some things in particular. That everyone in cohort 1 and 2 received the dose as expected. In cohort 3, 1 individual received a decreased dose of cells. They did receive the same volume and in the correct locations, but the dilution was performed 1 additional time, so that person, instead of receiving 20 million cells as 2 10 million cell injections, received 2 5 million cell injections for a total dose of 10 million cells 10 to the 7 cells. And that has been, a problem that's solved with the current lineage preparation, where the dose dilution doesn't have to be done in the way that this was done. It's an off-the-shelf product now. At the bottom of this in cohort 5, you'll also see that one individual, there's a caveat there. That individual had a very narrow lesion in the cord, and the neurosurgeon was not able to identify 2 places to inject with the suggested separation of 5 millimeters. So that person had a single injection of 10 million cells. So those are all of the kind of fine details. In the interest of time, I won't get into everything that's on here. I mean, this is basically very similar to what Linda had showed, but a couple things change. The enrollment window moves out from 21 to 42 days, which makes a dramatic difference, I'm sure, for the audience. Taking care of people at this phase, you're really starting to see people who are likely to be in an acute rehabilitation setting. In the thoracic trial, one of the things to highlight just logistically is a lot of those individuals were very close to their initial acute hospital stay. So this was a much more practical window to enroll people in. Everything else on there, basically, we've already said, and I would just say that it has the same structure of very intense, although slightly less than the thoracic, 1-year in-person follow-up with multiple INSCEs and multiple MRIs. Year 1 to 5, these individuals are seen in person. That was affected by COVID a bit, but we're moving back to seeing people again in person with MRIs through year 5, and then we'll follow them with phone follow-up through year 15. Now, paralleling what Linda had shown, this is a much larger trial, so the number of AEs being larger is not that surprising. 25 individuals treated. You can look through, you can see that the majority are mild to moderate in severity, but I did want to highlight a couple of AEs and SAEs. These are divided in the same fashion between related to OPC1. There was one individual at day 47 that developed a dysesthesia that actually resolved by 2 years into the study. For the injection procedure, one person developed a CSF leak that required moving from the acute rehab setting back into an acute hospital setting with a lumbar drain, and then that resolved. The person returned to rehabilitation and has been out. I actually see that person in follow-up now. And then tacrolimus had one adverse event reported at day 30, which was urosepsis. It's important to note that the degree of immunosuppression for this trial, it's using tacrolimus, but it's a fairly low dose in terms of the degree to which it immunosuppresses individuals. And that person was able to stay on immunosuppression and be treated with antibiotics. So immunosuppression was not held despite the fact that the person developed that complication. You can move to the next slide. I think this is really one of the most important take-homes from this trial. Clearly, it's a safety trial. No individual had deterioration in neurologic motor function. 64% of individuals had at least one level of improvement, so say C4 to C5 on at least one side of the body, and 32% of individuals had at least 2 levels of improvement on one side of the body. So 96% of individuals in this improved relative to their initial exam. We'll go on to just in the interest of time, we'll keep going. This is a bit of a busy slide, and as my background is in math and statistics, so I do want to say that I think that you have to take p-values in a study like this with a bit of a grain of salt. But I think directionally it does give some interesting information in terms of what seemed or what did not seem to be related or correlated with neurologic recovery or change in upper extremity motor score. So age not related, gender not related. Interestingly, AIS grade potentially is related, although I think if you did a Bonferroni correction on this, it probably wouldn't. I'm going to say a little bit more about AIS grade in a minute. The neurologic level of injury, not found to be correlated by p-values. The dose, number of days after injury, and manufacturing lot were all unrelated for these individuals. So Linda, you could move on to the next slide. Now, this is a bit of a breakdown getting into one of the things that I think is really the most interesting, and I hope we get to spend some time talking about this in the Q&A, is just the lessons learned from this trial. So this is the list of the individuals with the least neurologic improvement, and that might give us a bit of a guide about what might be done in a future clinical trial. One area of interest is that the individuals with C4 actually did not have as much improvement as some of the other individuals. That would raise some questions about what the breadth of enrollment would be for a future trial. Something that I think is a very strong learning lesson coming out of this trial is that this trial was done at multiple centers, and there was differences in practice in terms of placement of drains after the injury or after the injection procedure, right? Because the injection, that's actually worth saying something about that. The initial stabilization was performed, hopefully early based on all the data. And then this injection into the parenchyma of the cord with the stem cell derived product was actually done as a second surgery. When the second surgery was done, we noted that individuals who developed fluid collections, these are the last two individuals in the list, subject 23 and 17, both developed CSF fluid collections, and they were two of the individuals who had the least improvement across the trial. So going into a future trial, we would recommend that the neurosurgeon place drains in the wound bed after the surgery, and hopefully that would lead to an even more robust improvement across all of the cohorts. You can move forward to the next one, Linda. So another learning lesson, this was actually on the last slide, but I might as well take it here, is that the degree of lesion as characterized by the Asia impairment scale is one thing, but there's also potentially an opportunity for using what you might call a biomarker related to the MRI going into future trials. And in this particular case, if you look at the image on the left, these are MRIs, cervical MRIs, T2 weighted. What you're seeing at the blue arrow in the left image here is the highlighted signal, the enhanced signal is hemorrhage within the cord, and that's a known poor prognostic factor for neurologic recovery. If you look at the right side, the image on the right where the blue arrow is, that shows a bright CSF collection is what you're actually pointing at that circular area. And this is one individual where in fact, I think this is the only individual in the trial that did not have engraftment of the cells or a change that I'll talk about in 2 slides. You don't have to jump to it, Linda, but I think the teaching point coming out of this is that future trials should avoid enrolling individuals who have hemorrhage in the cord, and perhaps as more of a speculative statement, I would even suggest that we might think about trying to characterize people based, in addition to their INSCI exam, by neurologic findings on MRI as a biomarker to govern what we might expect for neurologic recovery for some of these individuals. You can move to the next slide. This is something that we talked about already. If you look at the middle slide, the orange arrow shows the CSF fluid collection. This is actually a common finding in individuals. Maybe not a common finding, it's not a rare finding. There's a bit of a debate in the neurosurgical community about whether to preemptively place drains to prevent fluid collections like this. As I mentioned before, in future trials, we would have a recommendation to place drains to potentially prevent these types of fluid collections. And if this is correlated with neurologic recovery or engraftment of cells, this could be something that could be useful, not just for this trial, but for other trials in the future. I think you could go to the next slide, Linda. I think one of the most intriguing things from this study, or intriguing findings from the study, is the MRI results. Linda mentioned that the cervical paper is currently under review. The thoracic paper is published. We're intending to publish a paper on, dedicated paper on the MRI findings. I'd have to say that I really appreciate Lineage's commitment to transparency of data, because availability of this kind of data, I think, is going to be extremely useful for the field to understand neurologic natural history for individuals who participated in these types of cellular trials. In particular, the highlighted finding in this is that in the majority of individuals who have this type of injury, these AISA and AISB level injuries, would be very likely to have narrowing of the cord at the level that you're seeing this sort of bright slice 16, and cavitation in up to 80% of the individuals. In this study, I mentioned one individual did develop a cavity. The other 24 did not. So this is dramatically different from what you would expect for the natural history with these levels of injury. And I don't think we can say that, you know, obviously all these individuals are still alive. So we don't know what that cellular matrix is, whether it's the OPCs directly or whether it's a secondary effect of having had the OPCs injected. But what is clearly true in the vast majority of individuals is that the natural history of cavitation has been altered, and there is a cellular matrix of some type present at the injection site. You can go to the next slide, Linda. We're running a little behind time, so I am going to just say I've said everything on this slide, and am interested in, you know, one key part of this. I think that future trials should consider trying to add augmentative strategies to the rehabilitation to see. We're doing the cellular product piece of it. We've typically pursued the course of rehabilitation, which is the rehabilitation that is the standard of care in the United States. But I think it's an intriguing question to wonder what would happen if we added some of the novel types of rehabilitation strategies that are being studied in clinical trials now. And with that, I will hand it off to Ed.
Great, thanks, Steve. Real quick, just a disclosure statement. I was an employee at both Geron and Asterias. I designed both of the Thoracic Trial and the Cervical Trial, and led the clinical teams that conducted those trials. Prior to then joining Lineage as the Chief Medical Officer for a year in 2019. And then since then, I have continued to do some occasional consulting work for Lineage on the OPC1 program. So I just want to put out there in full disclosure that, you know, I am inherently conflicted. So tough questions I'll defer to Steve and Linda. So we'll go to the next slide. So Lineage has continued to make some significant manufacturing improvements. So importantly, the cells that were used in both the thoracic and cervical trials were originally manufactured at Geron Corporation using an older process that was developed in the early 2000s. Since that time, Lineage has done considerable additional work on improving that manufacturing process. It now has a new ready-to-inject formulation, meaning the cells can be thawed right in the operating room and administered to the patient. Which eliminates the need for dose preparation in a bone marrow transplant lab, which was very cumbersome and incurred some logistical challenges. Lineage has also significantly increased the production scale of the cells, which is important as you get to later stage trials. And also in parallel has significantly reduced the impurities that were problematic in some of the early Geron attempts to get the initial IND cleared by the FDA. And those have now basically been reduced quite a bit, as we'll show you. There's been no reduction in the functional activity of the cells, so that's really important. So the potency of these cells has been maintained despite the process improvements. Along with that, there's been a number of new analytical and functional methods developed to characterize these cells, and also all of the animal-based production reagents have been eliminated. Again, these are all key steps for any program that's advancing towards late-stage clinical trials and hopefully commercialization pending approval. And there's considerable also patent protection on this that extends out as far as 2040. Next slide. So again, this slide illustrates several of the impurity markers that were present previously in the cells made at Geron. The GPOR is the Geron process of record. So those are the markers shown in blue here. And these are percentage of impurities. You can see that with the Lineage improved process that the percentage of these markers is significantly reduced across the board. So this is really important again as we contemplate getting into late stage trials. Next slide. So this is the device that I had designed back in the Geron days. It was intentionally designed to mimic the type of device that was used in all of the preclinical animal studies to evaluate the safety and efficacy of OPC1 prior to going into the clinic. But this design entailed a number of compromises. One compromise is that it's attached to the operating table and has a rigid needle that hovers over the spinal cord and then obviously goes into it. And it required that the ventilator be stopped while the needle was in the spinal cord, because while the patient is on a ventilator, the spinal cord is moving up and down, and you can't have that. The needle would be pistoning into the cord. So that's an important design compromise that needed to be addressed. In addition, this is a very cumbersome set with many parts. It's heavy, requires multiple sterilization trays and additional supply kits. And so it was really designed and intended only for use in early phase clinical trials and needed to be replaced with something more streamlined for later stage trials. And if we go to the next slide. So these are a couple of photos of the new device that Lineage has transitioned to. So this is a novel parenchymal spinal delivery system originally developed at the University of California, San Diego, and now taken into a company called Neurgain, with which Lineage now has a relationship. This is a very streamlined, compact device. It attaches to the patient. And so because of that, there's no need to stop the ventilator due to breathing motion because the entire system moves with the patient. And in addition, it has an infusion pump here that's much more compatible with the thaw or ready to inject formulation that Lineage has developed. Some of these formulations can be more viscous and you want to be able to inject them very, very slowly. And again, because this device is attached to the patient and rides with the patient, we can now slow down the rate of the administration of the cells to a period of several minutes, as opposed to previously where we were restricted to only about a 2-minute injection duration, again, due to the need to stop the ventilator. Next slide. So as I mentioned already, some of these benefits of better stability and control with the new system. It eliminates all the complex positioning. We don't have to stop the ventilator. It's really streamlined and easier for the surgeon to use. There is additional animal testing ongoing to demonstrate the safety of this. And there is now a device clinical trial planned in both subacute and chronic patients planned to start sometime later this year. Next slide. So with that, I'll hand it off to Brian Colley for next steps.
Hey, Ed, thanks very much. Hi, everyone. So I'm the chief executive of Lineage. So I've got the responsibility to continue the journey that's been described to you today. Linda, if you could advance one. What I'm going to share is that the work that we've been doing is intended to drive the program beyond where it has been so far. And including adopting a couple of improvements. So we feel that the initial data is definitely supportive of continued investigation. The question that I really want Lineage to answer is how much recovery is possible by utilizing these cells. So that's a comparative clinical trial that needs to be conducted. Ed described these improvements that we've made to the manufacturing process. Now, we have had some success with this. Similar efforts that we have done in a retina program recently led to a nearly $700 million alliance with Roche and Genentech. So we think the company is well positioned to be able to execute in this way on the manufacturing side. And again, as described, the validation of a new delivery system, which will start before the end of this year, is an exciting step because we think that the attributes which Ed described will be able to open a much larger number of clinical centers by using our new thaw and inject formulation. Overall, that data that we collect will support the design and ultimately the conduct of a larger study, which we intend to conduct. So overall, we really get to stand on the shoulders of giants with both the responsibility and the privilege of continuing this program forward. Linda, next slide, please. None of you need any help to understand what it means to a patient. My little story, acknowledging that I know less about spinal cord injury than every other person on this call, is that when I was talking with one of these patients and he described how important it was to just be able to gain a little bit of mobility and just gain a little bit more independence, really makes a passionate commitment for our organization and the continued work that Asterius and Geron and all the investigators involved with this program and supporters like CIRM were able to do. So in closing, there are many acknowledgements that go beyond this esteemed list here and of course the speakers today. So thank you all for your attention. And Linda, I think I could go back to you to direct Q&A for the group, if that makes sense.
Yeah, actually, Shannon, we don't see the posted questions. So I think you share those with us.
Yeah, right now, right
okay.
now in the chat, there isn't anything, but anybody's welcome to unmute themselves or put their video on and ask a question. I guess I can start. So with injection, is there any necessary like scaffold or like something to help it proliferate, differentiate appropriately, like attached to while you?
And why don't you take that one?
So the formulation of cells does not have another like biodegradable scaffold or something. The cells are able to migrate. They can proliferate, but it still is a relatively contained area. So there isn't any need in this iteration for any three-dimensional scaffold to hold the cells in place. Although I do think from time to time people do contemplate that as an option and how it could be beneficial. Although it does complicate development of products for sure.
I can imagine. Yeah.
I have a question. My name is Reza Asanian. Great talk, everybody. This was directed toward Brian or any of the other people. So the next step is going to be a randomized clinical trial. And I just for recruitment's sake for patients, having a crossover arm is really difficult in a trial like this. Has Lineage thought about what to do or how to speak to people who might think that a randomized control trial isn't ethical?
Yeah, it's a really important point. I think it's great that you brought that up. The next step is actually a smaller 5 to 10 patient device validation trial. While that study is ongoing, we're going to be working on addressing exactly that point, because from the perspective of a statistician, a controlled study with a sham procedure is mathematically preferable, but it also introduces some serious ethical questions about how to control this sort of study. There are precedents with FDA for various therapeutic approaches to be approved on a single arm study. There are methods and ways in which you can provide the agency with compelling evidence that there is a treatment effect without a control arm. But they certainly would prefer for that to be in place. So it's an open question because we know where their starting point is. But I think we're optimistic that we will be able to move them away from that, and perhaps by employing some of these other attributes in study designs and statistical approaches that help them get comfortable that there's a treatment effect without the double blind in the most controlled possible way.
Thank you.
Hi, I actually had a question, if it's okay. I'm Lana Zholdiv. I'm a postdoc at Gladstone Institutes. I think just to follow up on that question, have you considered using an adaptive clinical trial design, especially for a follow-up?
Yeah, if it's all right, I'll answer that as well. Absolutely. I think this is actually a very nice setting for an adaptive design. I think it's probably quite suitable. We haven't gone to the agency to propose one at this time, but that will be something that we intend to do. So I don't want to presume that I know exactly what the final study design will look like, but I think that this is a good example where having an adaptive design and being able to make a change on the fly. I like that as a CEO, simply because you can continue to have the sites open. One of the things that frustrates me with clinical trials is how long it takes to get a site up and running, even just getting the contracting. So I think having an adaptive design where we can continue to make adjustments on the fly, part of a statistical plan that's put in place a priori, of course, but I do think that there's a strong reason to consider an adaptive design for this particular patient population.
And maybe as a follow-up, what would be the pushback against trying an adaptive clinical trial design?
The question is very fair, but it's just from my perspective, it's the unpredictability of your engagements with FDA. Sometimes there are ideas that come from funny places with the panels or the advisors. So I think most sponsors just really look to try and cover all those bases, anticipate what they might be, and have those sort of in your back pocket ready to address. you know, the adaptive designs are a little bit newer. You know, they're more common than they used to be. So I think that provides a little bit of wind in the sails. But the agency's favorite phrase is to propose and justify. So if you want to propose an adaptive design because you think it's beneficial for some reason, if you just justify it and you have a compelling rationale for it, and oftentimes you can say, look, this is going to expose you know, fewer patients to an experimental therapy or more patients so that we get more compelling data. There are a lot of ways of presenting it that you can get, you know, kind of the outcome that you would like from those interactions.
And I'd just like to add, beyond this clinical trial, I think as a field in spinal cord injury and in rehabilitation medicine, I think this field is ripe for adaptive trial designs, and there are increasing numbers of discussions within both the spinal cord and rehabilitation community about integrating these designs. One of the major challenges is all of the upfront work that needs to occur and upfront costs, which can be prohibitive, particularly in an academic type study. But I really hope that study sponsors will move the needle forward in this direction, because I think it's a great opportunity for the right kind of study to really advance our approach to clinical trials. So that's my flag waving statement on adaptive trials.
I mean, I can ask yet another question. I'm always curious if, for example, rehabilitation, because it varies from site to site, from what, you know, the injured individual has had already versus what they have signed up for through private insurance, for example. So I guess considering the next steps, would rehabilitation or some kind of activity-based intervention be considered as a combination, or do you just ignore that part of the intervention altogether and just focus on the cellular therapeutics?
Well, I'm going to invite others. My view is if you're putting material in there, if you can stimulate it electrically, physically, that I think you'll get a better outcome. But Dr. McKenna, Ed, do either of you want please add to that?
Yeah, I think Stephen and Linda should answer this one. We did look into this quite extensively, actually, to try and figure out, should we be documenting what types and quantity of rehabilitation individual subjects were getting. And what we found is that, especially in the outpatient setting, after patients are discharged, there doesn't seem to be any cohesive standard of care that's universally applied. But that's all I'll say. Mila, turf to either Linda or Steve to address.
Steve, do you want to comment?
Yeah, I think it's a really good question. My observation, following a number of the individuals, I probably followed about a third of the people in the current trial, the full 30, and I'd say there is pretty significant variability in terms of what people are doing. Some people are signing up for these sort of specialized gym memberships, based if they have the financial resources just to pay for those out of their pocket. That said, I'll narrow down my thought on that, is that I do think that going forward in a clinical trial, it would make a lot of sense to have a ongoing active therapy strategy, right? And I think that a potential candidate strategy, I don't think this necessarily would win the race, but I think that I think there would be some criteria one would want to hold a potential ongoing supportive therapy to. If it is a randomized trial, I would give it to both parties, right? So one of the potential reasons why people might participate in the trial, if there were a control arm, is if they had access to an ongoing therapeutic intervention that's based on activity. So I think that would be something you would want to do. I think you would want a methodology that's potentially been evaluated in a clinical trial paradigm in the past. I'm not saying that MindMove, I have no financial relation with any company, but that's the one spelled with a Y for the I. They have set up clinicaltrials.gov registered clinical trials. with strategies to try to use electrical stimulation, FES, in strategies that are paradigmatically implementable in consistent ways across the upper extremity, right? So I think that type of intervention and FES-based plasticity, trying to get the cells to do something, like the analogy of, if you had a pile of bricks, would you leave it there, or would you like build something with it? I think that we haven't really explored some of the potential of what would happen if we do cellular therapies and then do activity-based plasticity enhancing strategies. And I think there could be some pretty clear strategies of what criteria you would apply for those. Again, I'm not going to say that any sort of strike me as being the winner of that race right now, but at the time that a trial happens, I think it would behoove any sponsor to, I mean, if you're going to spend hundreds of millions of dollars on, your clinical trial project, investing some of that in having a therapeutic intervention on the back end to maximize the potential of the cellular intervention, just makes a lot of sense to me.
And I'll just add, I agree with everything that everyone has said, and also to say that the preclinical evidence supports the use of rehabilitation in conjunction with a cell or drug-based strategy, and that I think that rehabilitation interventions, as Steve mentioned, have advanced in terms of activity-based therapy. Locomotor training is the standard of care, which it was not at the time this study was initiated, but it has become a standard of care. And I'll also say that other sponsors are integrating rehabilitation within their study protocol. So I think that's the direction the field is going, and I think it's the direction it needs to go to really optimize success, because rehabilitation is definitely a confounding or an effect modifier in terms of therapies geared towards remyelination or regeneration.
All right, any last questions? Well, thank you guys so much. We really appreciate it. And I'm sure there's going to be many trainees tuning in after this with the recording. So I'm sure maybe we'll have some reach out to you guys in the meantime.
Great. Thank you so much. And again, thank you for the invitation from AAP, and we're glad to be the final presenters in 2022.
Thank you so much. Have a great night, everyone.
Thanks, everybody.
Thank you so much.
Bye-bye.
Bye-bye.